As we learn about proteins, you can see we've got a really long list of things that this is going to help us review. Lots of concepts come together for these proteins that we're going to be looking at. So we'll just take some highlights of them. Alcohols, that's that OH group. Thiols, we're going to see that's an SH group, very similar but with a sulfur. Carboxylic acids, we already mentioned on the first page. We will be using some pink to clue us into those throughout the day. Amines, we've seen those as well before, and we'll use some blue to key us into those amine groups. We will also see two functional groups that we saw earlier in the semester, chapter 13. So this was chapter 13, amides and esters. And just a reminder, with both of those, we said that those were linking functional groups that could be broken in half by hydrolysis. So we'll be seeing that again. Other reminders, we will be talking about how amino acid side chains and backbones get held together by intermolecular forces in this chapter, so remember those attractions. We'll be talking about polar molecules. Back in chapters 4 and 7, we used the molecular dipole for that. We won't use those so much here, but polar, in these larger molecules, we're going to look for nitrogens and oxygens. Those tend to give us polarity. Non-polar molecules, remember that was when we see carbons and hydrogens exclusively. Those are sharing electrons really evenly. And then we'll talk about some intermolecular forces, dispersion forces, hydrogen bonding. Remember, hydrogen bonding is we're looking for NH, OH, and FH, and then N's, O's, and F's. We won't actually see any fluorines in this chapter, so really just those N's and O's are important. And then ion dipole, that was the strongest intermolecular force we've learned so far. Remember, when we have ions, then we have either a positive cation or a negative anion, and those lead to strong attractions. A couple other reminders. Again, chapter 8 and 13, we saw some glimpses of those. Amides, hydrolysis reactions, review those. Protonated and deprotonated compounds, so those will be some terms that we'll see again. And then some other colors and things that we will use. We've been using throughout our biomolecule sections the purple for the cell membrane. If we have energy, we're talking about ATP. Hello, ATP. We will also be focusing a lot on the side chains of our amino acids, and I'll be circling those in green throughout today. Proteins as a substrate or as food will be a dark highlight. And then at the end of the chapter, we're going to be seeing a little bit about enzymes, because enzymes are a type of protein. And in those diagrams, our enzymes will be orange. Our cofactors, which you'll learn more about, will be pink. And our inhibitors, again, we'll learn more about those, will be blue. Okay? So as we begin here, I'm going to do just a real quick sketch of what a protein might look like. Okay, so we're going to see something called alpha helices. Those have this coil look. And this would be a long polypeptide that I'm drawing. Okay, and so we'll see alpha helices curled up. We'll see beta sheets over here. This would be alpha, alpha, and then some beta sheets here. And in this polypeptide, what you're going to see is first of all, in the backbone, there's a whole lot of these amide functional groups holding amino acids together. So all kinds of amides as I go through my protein. All right, and these are not to scale and not exactly in the right spot, but lots of amides holding my amino acids together. And then in between those, we have lots of side chains. So remember my side chain of amino acid. We'll go into more detail on these, but these basically just kind of protrude off of my amino acids into the spaces in between some of those coils. And so I've got lots of side chains throughout my molecule. And those side chains are going to be really important. All right, now those side chains are going to be held together by intermolecular forces. And so when I'm holding a alpha helix together, these are going to be what we call secondary attractions. Or when I'm holding amino acids together within beta sheets, also secondary attractions. I'm going to add a couple more amino acid side chains on here to help me out later. All right, so second, we're going to talk about primary structure is just building this polypeptide. Secondary is when we hold a helix together or hold some beta sheets together. Tertiary attractions are going to be when side chains hold two helices together. So this will be a tertiary attraction. Or say I have my helix attracted over here to the beta, that would also be tertiary. And then finally, if I can grab another color here, just a second. So I can have a completely different polypeptide. Maybe it's got a bit of helix here, okay? So this would be polypeptide number 2, and up here I have polypeptide number 1. And so this polypeptide would be constructed in the same way with side chains all over it. And so our final type of attraction that we're going to see is that these two can actually be attracted to each other. And that would be what we call quaternary structure, where the side chains of two different polypeptides are attracted to each other. So that is kind of a big picture sketch that we will come back to throughout the chapter in some different formats. As far as our structure and properties and function go, I'm going to zoom in a little bit here. Some of the things that we will notice in our proteins is that we've got, zoom in here, side chains on amino acids are going to be very important in our structure. We're also going to see, like we were just looking at, primary, secondary, tertiary, and quaternary structure. And those polypeptides end up folded into some different types of arrangements. So I'm just going to say those polypeptides end up folded up. As far as properties go, there are so many different properties of proteins. We will see just a few. And then other things we'll talk about with the properties is we will talk about regions of the polypeptides, the proteins that are polar or non-polar, or acidic or basic. So those will be some of our key terms that we're going to use as we talk about the properties. As far as the functions, again, properties and functions go together. So we again have so many of those different functions, but a couple would be for food and also for structure. And we'll see, as one detailed example, we'll be looking at enzymes as one function of our proteins. And then finally, as far as reactions go, the main one that we're going to look at today is going to be hydrolysis. So that gives us a big picture glimpse of what is coming up with our proteins. And just zooming out a bit on those sketches and things that we wrote down, I just want to briefly add on another version of how we can see and think about these proteins. So again, if you check this out in color, it will look a bit different. But you can see here this primary structure. We're going to write that as 1 degree. Secondary is where we take our straight strand and start to fold it up into helices or into sheets. Tertiary, we take those and coil them together. And then quaternary, that would be where we had the one strand coming around, and then we had a whole different separate strand coming around in the last picture. And so we'll have definitions of these as we go, but a couple of just new terms over here. The backbone of the amino acid would be here, and I tend to read it NCC, NCC, NCC. That's that amine, amine, middle carbon, carbonyl, amine, middle carbon, carbonyl. So that shows up as my backbone. The side chains, we have seen those before as well. We can't really see them easily in this picture, but just adding on where those different important types of bonds would be. In primary structure, we're going to see the amide bonds show up, linking my amino acids together, and then we start folding. So as we go this direction, we are talking about folding up my protein. And you would see intermolecular forces holding those beta sheets together, holding that alpha helix together. That's my secondary structure. In tertiary structure, you're going to start to see, say, a helix attracted to a helix, and so that would be, again, intermolecular forces. And then in quaternary structure, we find attractions between two polypeptides, and that's going to be what we see later in our definition of quaternary. Now, if we can fold, it makes sense that we can do the opposite. And so we will talk later today about unfolding. And unfolding, the term for that in proteins is denaturing. Denaturing. So we can fold, and then if we have the right conditions, we can unfold. And that may or may not be something we want to do. So that just adds on to our previous page.